Hello, everyone. Thank you for joining us for today's webinar, Understanding Batten Disease Pathogenesis, presented by Dr. Jonathan Cooper, Professor of Pediatrics, Washington University School of Medicine, St. Louis. My name is Xavier Gutierrez, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Now, before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. And if you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Cooper. I will now turn the presentation over to him.
So hello, everybody. It's a pleasure to be with you today and to have the opportunity to tell you some of the work that we've been doing to understand a group of diseases called Batten disease. And the main reason we're doing all of this work is because we're trying to come up for therapies for these fatal inherited disorders. So first, I'd like to introduce the diseases to you. They have a long scientific name, which is Neuronal Ceroid Lipofuscinoses, but typically we refer to them as Batten disease, which is actually an umbrella term for a whole family of related diseases. They're named after the gentleman you can see there in the top corner of the screen, Frederick Batten, who was an eminent British pediatrician who described these early in the 20th century. Now, they are a whole group of diseases that have been put together. Initially, we thought there were only 4 of them. Now it's recognized that there are at least 14 and maybe more of these diseases that have been grouped together on the basis that they are all profoundly neurodegenerative. All but one also involve degeneration of the retina, but they have very characteristically this buildup of autofluorescent storage material inside the lysosome. And that feature of material building up inside the lysosome is what makes them what we call a lysosomal storage disorder, because things are building up within the lysosome that shouldn't be there. And I'll explain in a moment why that happens. Clinically, these are diseases that start at very different ages. They can start from just after birth all the way through to an adult onset form, but they share certain common features, being that affected individuals will suffer visual failure, progressively worse seizures, and a horrible and relentless decline in both cognitive function and motor ability. And sadly, all of these are fatal diseases, and that's why we're working very hard to both understand them and come up with therapies for them. Now, they are grouped together largely on the basis of that storage material that builds up, and it's very easy to see. We can simply shine ultraviolet light upon sections so we don't even have to stain them. And you can see here lysosomes that are chock full of that material that should have been degraded that is built up inside them. Now, these are all inherited diseases. Each of them is caused by a mistake in a different gene. They're what we would call monogenic, in that it is a single gene defect in each form. And depending on which gene carries that mutation determines which age and how fast the disease progresses. Now, with only one exception, they're all inherited in a classic autosomal recessive fashion, meaning that both parents are carriers, they are heterozygous for these mutations, and every time they have a child, there is a 1 in 4 chance that that child will inherit the mutation from each of the parents, and having 2 copies of the mutated gene means the child will unfortunately have the disease. Now, as I said, they fall into the category of diseases that are called lysosomal storage disorders, and I'd just like to take a short moment to explain what those are. So if we look at a cartoon cell containing lots of different organelles, our main center of interest is the lysosome. And we can think of the lysosome very simply as being the cell's waste disposal center. It contains machinery. You can think of it as either scissors, I prefer to use the Pac-Man analogy, that are going to break down proteins that are no longer wanted inside the cell so that they can, perhaps more accurately, rather than simply waste disposal, be recycled so that they can then be used to make further proteins. Now, what happens in all of these diseases is that process of waste disposal breaks down. There's a failure in the recycling, and we get a buildup of material or stuff inside the lysosome. And that's the autofluorescent material that I showed you on the previous slide. How that actually happens, if we look at a cartoon version of the lysosome itself, a double-walled vesicle inside the cell. Inside it, there are multiple enzymes, or Pac-Men as I've used here. Their job is to degrade material that ends up in the lysosome and get rid of it. Now, what happens in all of these diseases and all of the lysosomal disorders is that those enzymes become dysfunctional. And what happens, it's rather like if a waste disposal unit at home were to go wrong. If that fails, stuff would simply build up inside that lysosome or inside your waste disposal unit till you end up with one that's full. And that is precisely what we can see when we look down a microscope. It's also what you can see in our lab logo. We work on a group of these diseases, all of which affect children. So we are called the Pediatrics Storage Disorders Laboratory. Now, there happen to be about 70 different diseases like this. And while they might each individually be very rare, and we're talking like 1 in 200,000 maybe, put them together, all of those, that is some more like 1 in 5,000 of children will have one of these diseases. And given that they are fatal, we actually really want to do something about that and come up with therapies for preventing these children from dying. So what actually happens in the different forms of NCL, to use the abbreviation that we typically use, is that we have genes that are mutated that are actually in different places inside the cell. So many of them are an enzyme, or that Pac-Man. And that's true for the infantile, the late infantile, the CLN10 congenital form, and probably for the CLN5 form, is that we have a mutation in a gene that should code for an enzyme. Because of that mutation, we get no enzyme made. There are also forms of NCL which are a mutation in a quite different sort of protein. So that wiggly line is how scientists like to show a transmembrane protein that was sitting in the wall of the lysosome. And that's true for the juvenile CLN3 form of the disease and also for CLN7. However, there are other forms of NCL again that are mutations in transmembrane proteins that are coded elsewhere inside the cell. So either in the Golgi apparatus or in the endoplasmic reticulum, as is true for CLN6 and CLN8. And by mechanisms which are only really just becoming apparent, this has a knock-on effect upon the lysosome. So the story is that effectively we have different protein deficiencies that take places in different places inside the cell in each of the forms of the disease. So we're beginning to see that they're not actually that similar to one another as you might think. So I'd like to emphasize that point, that we do have quite different forms of Batten disease, or NCL. So some of them fall into that enzyme deficiency category, typically CLN1 and CLN2. Others fit into that transmembrane protein deficient form, prototypically CLN3 disease. Now, given that we have diseases that are caused by mutations in genes that have very different functions and are in different types of proteins, even in different places inside the cell, it wouldn't necessarily be a surprise that how things go wrong, which is another way of explaining the word pathogenesis, would be rather different between these different forms of the disease. Equally, when it comes to devising therapies, the implications are quite different. For those enzyme-deficient forms, we can rely on the mechanism of cross-correction, in that there are receptors sitting on the surface of cells waiting to catch any enzyme that we could deliver to a brain, so they will be taken up to cross-correct the neighboring deficient cells. That isn't a possibility for a transmembrane deficient form of NCL, and we're largely looking at trying to understand what are the downstream effects of those mutations and come up with other ways to try and block those effects and hope that they will be therapeutic. Although, as we're discovering now, gene therapy is holding a lot of potential for those transmembrane forms of the disease too. So the foundation on which all our work is done is working in a series of different animal models. And our challenge is to know and to understand those different models, be they a knockout mouse, or be they a naturally occurring larger animal species like a dog or a sheep, is to try and understand them. Now, they are all good for particular purposes, but they equally have their limitations, and we have to remember they are purely a model for the human disease. But what we like to do is to ask some very simple, or as I would call them, chicken and the egg questions about these models and use that to answer our understanding of the disease and push it forwards. So what are the landmarks? What are the things that we actually wish to measure in those? Is it pathology? Is it tissue culture? Is it biochemistry? Is it behavior that will be most informative? And effectively what we're doing is defining how these diseases start and how they progress. Now, the reason for doing that, while it's interesting from a scientific perspective, is actually with a very focused purpose, which is to define where and when and how we can deliver our therapies to be most effective. So the way that we do that in my laboratory is understanding largely pathology. And that can be pathology in mice, or it can be in those sheep or pigs or dogs, as we shall see later. So our starting point is the brain, and here is the brain of a mouse. And we prepare a series of different sections and stain them in a variety of different ways to learn things and find particular indicators of pathology. Often before we prepare those sections, we have done behavior on those mice, or we may have taken cells from other mice and grown them in tissue culture to ask particular questions, or we may have looked at gene and protein expression. But if we turn our attention mostly to those tissue sections, we're wanting to extract numbers. We use a series of different microscope techniques and a series of different packages to extract numbers about how the structure in those brains has actually changed. That can involve doing things like thresholding imaging analysis to ask how much of something is there. For example, we might measure how much of that storage material is present, or how many of the indicators of disease might have disappeared. But a lot of it also comes down to doing stereological, unbiased stereological measures of structures in the brain. And stereology, as we'll see, is a very important method, particularly in brains that have changed their shape as a result of disease. And all of that work we do is blind to treatment and to genotype, so that the people who are doing these studies don't actually know which animal is which. And it's only at the end of our studies that we crack the code and sort the data into different groups and try to determine whether our treatment may have had an effect or not. So just to emphasize why stereology is important, obviously the main focus of our disease that we're interested in is the brain, and the brain contains millions and billions of neurons, and counting every one of those is not feasible, even within just one small part of the brain. We become bored doing that, our counts would become inaccurate. So what we do effectively is to sample a known fraction of that tissue, and based on that estimate of what we counted in a small bit, we then extrapolate up to what was in all the bits we didn't waste our time counting. Now, for that method to actually work, it's crucial that the sample that we take must be completely unbiased. And that's a term for sampling structures within the brain. And it's as cells get bigger or the brain might shrink, our probability of sampling them would change. So we have to come up with a method that will overcome that. That method is done by superimposing a grid that's regularly spaced on top of sections that are themselves regularly spaced of known thickness. And then we ask simple yes or no questions in a small box called a dissector frame. And from that, we can get rapid and efficient ways to obtain unbiased data from brains that may have atrophied significantly, or in which the cells may have either shrunken or got bigger because they're filling with storage material. And that's why stereology is a very important method to use. So the sorts of things that we're looking at, obviously these are profoundly neurodegenerative diseases. So we have a lot of neurons that are dying. We have that buildup of storage material. We also have a lot of inflammation within the brain in terms of reaction of astrocytes, a process called astrocytosis, something similar of microglia within the brain. And we also have synaptic pathology that we know takes place. Now, many of these things happen in a whole range of other diseases too, perhaps with the exception of the storage accumulation. But our interest is in actually looking at what is the relationship between these events and how is their timing different in different brain regions. And all of that is in terms of being able to target our therapies. Now, as a result of many years' work, we know that these diseases effectively end up at a very similar endpoint, but they may take a different path to reach that pathological end stage. And that's exactly what we've been trying to get to the bottom of. The first thing that we discovered is that not all neurons respond the same way in these particular diseases. So some neuron populations are more vulnerable, and by counter-argument, some are actually more resistant to the effects of disease. We first discovered that for populations of GABAergic interneurons, which help control brain activity, and it's not a surprise that they might be defective in a disorder where we see seizures. Perhaps more surprisingly was that the relay of information from the thalamus up to the cortex was affected far earlier than the cortex itself. The dogma was that this was a cortical disease. It may end up being so, but largely secondary to effects within the thalamus. There is also a theme that the larger and more complicated neurons are, the more likely they are to be vulnerable, and examples are laminar 5 pyramidal cells or Purkinje cells within the cerebellum. And recently we found another good example, which is the selective pathology that we see early in the disease within the spinal cord. Now, obviously, if different things are happening in different places, this has big implications for where and when we should be delivering our therapy to have the greatest effect. Now, an obvious question is what's actually causing this selective neuron loss? And the first finger of suspicion pointed at that storage material, perhaps largely because it is so easy to find simply by shining ultraviolet light upon its action. But when we measured where and when that material was building up, it did not have any direct relationship to which neuron populations were dying, suggesting that the two events are not actually linked. And what we think is that as the disease progresses towards a lot of neurons dying, that this storage material is effectively a non-crucial byproduct as part of the disease. It's a disease marker, but it's not the most important event. And that raises the question then, what else might be important if it's not the storage material? Now, it turns out that one of the best and earliest predictors of neuron loss is something that I've already mentioned, which is that glial activation or inflammation that takes place within the brain. The surprise was that this was happening in very small patches of the brain very early in the disease and was actually happening before neurons were dying. And it turns out that this is actually a very accurate predictor of where subsequently most neurons will go on to die and those that die first. And it turns out to be a much better predictor than that buildup of the storage material. One of the first places we defined that properly was actually in a sheep model of the disease and discovered that it was present actually very early in the disease. In those sheep, it's actually present prenatally. And that, of course, raises the question whether this is present in many other similar diseases now. It raises the question as to whether this is a positive or negative event in disease progression. If it's something that's a positive, potentially regenerative thing, it's something we might encourage. If it's a negative event, it's something that we might wish to try and control or damp down. So we've been looking at that and asking recently, what's the contribution of these glia and that inflammatory process to the disease progression in multiple forms of the disease? This comes around because in a regular healthy brain, there is an intimate relationship between the neurons, the astrocytes, and the microglia that is crucial for normal brain function. And whilst most of our attention has been on the neurons as the culprits in this particular disease, it's entirely plausible that problems within the astrocytes or the microglia could end up with causing a cascade of ever-increasing problems that end up with neurons dying. Now, it's entirely plausible that astrocytes and microglia could be affected because they too express those genes which are defective in these diseases. So we've been asking very simply, how is each of these different cell types affected in the disease, and how are the interactions between these cells altered? And we've been doing this comparing across the three major forms of the disease, CLN1, CLN2, and CLN3 disease. Now, the way that we do this is by going cultures from early postnatal mouse brains. And we have methods where we can grow cultures of just those astrocytes, or just microglia, or just neurons, and then ask very simply, how is each cell type affected in each of the different forms of NCL? Once we've then defined that, we can then start mixing and matching. And in co-culture systems, we can grow different combinations of astrocytes and microglia and neurons and mix and match healthy or mutant cells and see how they interact and influence one another. So what I'm going to do is talk through some of the results we've got from first CLN3 and then CLN1 mice and show you what we actually found. So this work started, and I'm going to talk first about CLN3, was that looking at the brains of these mice, we found that whilst astrocytes were becoming activated to the point they were upregulating the expression of a marker called GFAP, they weren't actually changing their shape normally. So at the top there, we can see a wild-type healthy mouse in which you see a palely stained astrocyte that looks a bit like a firework burst going off. In the far right there, you can see cells that have become fully activated. They have a much larger cell body. They stain much darker and have very few but thick processes. In the middle, we see a CLN3 mouse in which, yes, it is darker and more brown, but it hasn't changed its shape fully. It's as if it's got started and then got stuck partway along through that process. Now, that's something that we also see in our cultures from those mice, in that when we stimulate the cells, they can't change their shape as readily as healthy cells would. And that's because they have a series of cytoskeletal defects, and that has a knock-on effect on their ability to migrate into a scratch assay, done again in tissue culture. Not surprisingly, because they have cytoskeletal problems, we also see that they have a series of defects in their ability to secrete chemokines and cytokines, and that is more pronounced in astrocytes than is seen in microglia, which have no problems in phagocytosing things we give them. Those astrocytes are certainly very severely affected. They have trouble taking up glutamate within a culture, which would obviously have implications for excitotoxicity. They also have problem in generating spontaneous calcium waves, which is how astrocytes talk to one another, suggesting that this process may also be impaired in CLN3 disease. We also looked at the neurons. If we grow neurons from CLN3 disease mice, we see that they, whilst they don't die, they do have very particular structural defects in their dendrites, which may well be related to their ability to communicate with one another, and an interesting phenotype with their axon initial segment. The interesting part of this particularly came when we started growing neurons and glia together. So this is growing a mixed culture of the neurons and the astrocytes and the microglia together. And what we found was that CLN3 mutant cells, those mutant glia, can actually kill neurons. They will harm healthy neurons, but they actually kill mutant CLN3 neurons. But if we then grew healthy glia together with sick neurons, this was actually capable of protecting the phenotype of those sick neurons, suggesting that if we could actually treat glia, we may be able to help rescue some of those neurons. So armed with that information, we then conducted very similar experiments in CLN1 disease, perhaps foolishly thinking we might find something very similar. But in actual fact, we found quite a different story. What we found was that microglia in CLN1 disease were actually far more activated even before we stimulated them. But when we did, they responded perfectly normally, which is quite unlike CLN3. What we did find, though, was that CLN1 microglia appear to have an impaired ability to eat up small latex beads, so impaired phagocytosis. Turning our attention to astrocytes, they too, CLN1 astrocytes, were more activated basally before we even stimulated them. But again, they had a normal cytoskeleton and responded normally to stimulation. Again, two things that CLN3 astrocytes had problems in doing. We also find that they had differences in their ability to secrete chemokines and cytokines, but quite different to those we saw in CLN3 disease. We then looked in more detail at the astrocytes and found, surprisingly, that over time, there were many fewer CLN1 astrocytes in our cultures, suggesting that they were simply dying in a dish. They appear to die by a process of apoptosis, and that parallels some data that we have from the brains of these mice in vivo. We also found that there were particular calcium signaling abnormalities in those CLN1 astrocytes, and that they had higher levels of cytoplasmic calcium, and this was being released from lysosomal rather than ER stores. We then looked at the neurons, the CLN1 deficient neurons, and we found that unlike CLN3, they too were vulnerable in a dish. So fewer neurons survived, a bit like the CLN1 astrocytes were dying over time. So were the CLN1 neurons, but this was most pronounced for the inhibitory neurons, just as it is in vivo. Again, this appears to be by a process of apoptosis, with the surviving neurons looking sicker and having a smaller cell body. They also have a range of dendritic abnormalities too, but they're different to those we saw in CLN3 neurons. And there are a series of synaptic defects too. We then started doing those co-cultures, and this is where the story became particularly interesting. Firstly, if we grew CLN1 astrocytes and microglia together, while both cell types were inherently more activated, when they were grown together, they became more activated still, as if they are cross-talking or priming one another. If we grew CLN1 astrocytes and neurons together, we found that the astrocytes would impair how those neurons appeared, but didn't actually kill them. Whereas if we grew CLN1 microglia and neurons together, those microglia were capable of killing a subset of the neurons, and the remainder looked sick. However, when we grew everything together, astrocytes, microglia, and neurons together, we found that that process of cell death was far more pronounced. It appears that the astrocytes and microglia are combining to exacerbate the effects on neuron survival. So put together, we actually have a whole new perspective on the disease. One of the important things I'd like to leave you with a message of is that these diseases are not the same as one another. In both CLN1 and CLN3 disease, we have dysfunctional glia, but they are dysfunctional in quite different ways. So they're actually really different diseases at a cellular level. Another very important thing to come out of these studies is that while we have been previously thinking purely of treating the neurons, we really should be thinking of also treating the glia when we are devising therapies. So these are cells that clearly are dysfunctional and appear to be contributing in a big way to the disease process. And so we are now attempting to block these sorts of effects with a range of different compounds. So moving on to another section, I want to give you another example of how pathology is very useful in terms of informing the therapeutic efforts that we are trying to do. So I said earlier, we've been asking which parts of the brain are affected. Well, that's really important because we need to put the therapy in the right place. And we're actually finding a whole series of new and perhaps surprising targets, both within the nervous system and also within the rest of the body. And a good example of that I mentioned earlier was that really was a great surprise to find that there was pathology within the spinal cord very early in the disease in CLN1 disease mice. We know that that happens not just in mice, but we know there is also spinal pathology in human CLN1 disease. And we know from our studies that this appears to contribute to the disease outcome. It also appears to be present in many of the other forms of NCL too. So we think that this is an important problem to tackle. And we've been trying to go about doing this by doing gene therapy to provide a functional copy of the gene to deliver the enzyme that is missing. And I'm going to go through a series of data that will show you how and why we did those studies and how we came up with these effects. So we were treating CLN1 mice with gene therapy into their brains, and we found that we were able to largely rescue pathology wherever we put that therapy. So effectively, the forebrains of these mice had been cured. However, those mice were still dying, telling us that treating the brain itself wasn't sufficient to have a full cure of the disease. So that suggested that we had actually missed something and that there might be some important pathology elsewhere that we needed to target too. And it turned out that where we had missed was actually that there was a massive amount of unexpected pathology within the spinal cord. We first saw that that was present within the human disease and then went looking within the mice. And sure enough, it was there too. And it was there in the spinal cord before pathology started within the brain. So we then started trying to treat that. And so in addition to directing gene therapy into the brain, so intracranial, we also directed our gene therapy into the spinal cord intrathecally by means of something like a lumbar puncture. And so whether we look at the amount of storage material that's present, or whether we look at microglial activation, effectively where we put that therapy, we managed to cure the disease. So we have an untreated mouse at the bottom. We have a wild-type mouse that has very little inflammation. We have an untreated mutant mouse that has inflammation in both the brain and the spinal cord. If we treat the spinal cord alone, so an intrathecal treatment, we effectively fix the spinal cord but don't touch the brain. The disease is still present there. If we direct our therapy to the brain alone, we are able to treat that pathology there but don't touch the cord. However, if we treat both sites, we can effectively prevent that pathology from taking place in both sites. Perhaps most importantly is that this didn't simply have an additive effect, but was synergistic. This is looking at behavioral data, and we can see that the ability of the mice to stay on a spinning rod, something called a rotor rod, is much, much better when we treat both sites. But perhaps most importantly was that the lifespan of those mice was effectively tripled. If we look at the blue line in the lifespan curve there, an untreated mutant will die around 8 months of age. If we treat just the spinal cord, that will go out to about 12 months. We treat the brain alone, that will go out to about 16 months. But if we actually treat both the brain and the spinal cord, we're able to get those mice now to live nearly 3 times as long as they would have done had we not treated them. So that is by far the best therapeutic efficacy that we have seen so far in this particular disease. And all of that approach came about purely because we had discovered there was pathology in an unexpected site. And we are now looking at ways to try and improve this further by targeting the places where we think there is additional pathology taking place within the rest of the body of these mice. So I've talked a lot about mice, and mice have been very useful and very important for our experiments. But it's important to remember that a mouse brain is not really much like a human brain. It's rather small and it's rather simple. And we need to find ways to scale up our delivery to a much larger and much more complicated brain. And I've already mentioned the use of some larger animal species. These were mostly naturally occurring, but now there is the possibility to generate such models by CRISPR-Cas9. These larger models give pathology that's much more like the human disease and are a very effective testing ground for the practical issues of how to deliver therapies. So there exist naturally occurring and genetically engineered sheep. There also exist genetically engineered pigs. And I'm going to give you an example of some of this work that we did in a naturally occurring model of CLN2 disease that led to the first FDA-approved therapy for any of these particular diseases. This work was all done in collaboration with a company called BioMarin, and they were working with a TPP1 deficient dachshund, so a naturally occurring model that has a mutation that gives it a disease which is effectively CLN2 disease in a dog. So they were delivering human recombinant enzyme into the ventricles, the fluid-filled spaces in the brains of those dogs. And we, at the end of that study, got a look at the pathology and looking at the storage, looking at the inflammation within the brains. And what we found was there was a remarkably positive effect upon the pathology in those treated dogs. And that was true even at the end stage of the disease. Those effects weren't confined to pathology, but it effectively improved disease in these dogs. They had a much longer lifespan, a much better quality of life, and improved cognitive and neurological performance. So that work, which we were involved in, was the testing ground that led to a successful clinical trial for this approach in people, where the same enzyme was being delivered through a port into the ventricles. And this subsequently, at the end of a phase 1/2 clinical trial, it was shown that this was effective in stabilizing the disease at the stage that it was given. And that led to the FDA approval of Brineura as a product which is now approved for the treatment of CLN2 disease in children. And just to emphasize, all of that came from following the same pathway that we have talked about in this lecture. Understanding the pathology, understanding where it is, and being able to target it to the right place at the right time to have a treatment effect. Now, it's great that we have got to that point for CLN2 disease, but we need to make effectively the same progress for all of these different diseases, for all the forms of Batten disease, so that children who suffer from the other forms will have the same hope of a treatment in the future. So our challenge is actually to turn experiments that we do in our animal models, ultimately into clinical trials, and for those to become treatments. So in finishing this lecture, I've shared with you some of the basic principles of these diseases. I hope you have seen that these diseases are not simply the same disease happening at different ages, but they have quite different underlying ways in which the cells in these brains are actually affected. And it's also true from a clinical perspective. If you talk to clinicians who see these children, they are not quite the same disease, but there are particular differences. It's important to understand that not all parts of the brain, or indeed as we're learning the body, or even cells within those different parts of the brain are affected in the same way or to the same extent. It's also important to understand that whilst we have animal models, the pathology can be quite different between those species. So really understanding them is very important. And understanding that gives us the vital information of where to put the therapy and when to deliver it so that it can be most effective. And as we're learning, some of those places we're going to deliver it may well be in the future outside the brain so that we can have a complete treatment effect. So thank you for listening. I'll be happy to take questions that you can send in via email. I have to close by acknowledging the work that everyone has done. This is very much a team effort. I'm very fortunate to have a group of very talented people in my lab at WashU, but we have worked together with many people in many places all around the world to make this happen. We've been fortunate to be funded by a whole series of organizations and work together with some companies to move this work forwards. Thank you for listening.
Thank you, Dr. Cooper, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that this webcast will be available for on-demand viewing through June of 2019. And as a final reminder, our speaker will follow up with any of your questions you've submitted via email. That's all for now. Thank you for joining us, and we hope to see you again soon.